Morning everyone. In this class we are going to learn about evaluation of crude drugs. So before we enter into this class, what is the crude drugs? As we all know in this previous hours, and how to evaluate the ingredients which is present in the crude form of drugs. Okay. So what is crude drugs? Which is obtained from the natural origin, which is nothing but our plants, animals, minerals, and microbes and marine environment, and direct parts of exudes or products. So these are all the crude drugs and unprocessed one. Crude drugs are the unprocessed one, and which is only collected and dried storage and packaging. So these many methods are included to making a crude drugs. Okay. What are all the examples of crude drugs? Here it is: acacia, agar, benzoin, beeswax. So beeswax are the naturally occurring compound. So we no need any preparations or anything. So cinchona, which is derived from the bark, cinchona tree bark, and cinnamon also we know, and we are using culinaries in our food makings, and so ginger as we all know. So these are all the commonly occurring plant products. So these are all used as a food additives as well as crude drugs. Why we are call it as a term crude drugs means we don't know which compound is especially just going to heal some properties, so some medicinal properties, so we don't know anything specifically. So in that condition we are collectively call it as a crude drug. Okay. So definition of drug evaluation may be defined as the determination of identity, purity and quality of drug. So how we are using the term, this is the drug. So after passing these categories, which is identity, purity and quality. So after once we are producing one crude drug, we need to pass these kind of categories, then only we may call it as a drug. First one is identity. Identification of biological source of the drug. The drug which is derived from which part of the plant, which is which part, so which is the biological source of the drug. For example, carrageenan, which is from seaweed. Okay, so that is the source of carrageenan. And purity, the extract of a foreign organic material present in the crude drug. So that is the purity. So we are collectively call it as a crude drug. So from crude drug, which organic material we are going to extract from the crude, that is a purity. And the final one is quality, the quantity of the active constituents present. So before that, what we are saying, the purified form of exact compound which is derived from the crude drug. So in case of quality, how much quality, that means how much quality of the purified compound. Okay. And what is the importance of evaluating the crude drug? Why we are going to do this crude drug evaluation, and what is the importance? So these are all the things we are going to see in this slide. Okay. To find out the standard and substandard drugs. So after completion of these process, what we are going to know, the standardization. So how standard the drug. Okay. Then to check or to report the presence of impurities. Is there any impurities present in it, or is it completely pure? So these are the things we are going to check in this category. So substitution and adulteration as result of careless ignorance and fraud. So if some of the manufacturers they are doing adulterations, substitutions, and some careless ignorance mistakes. So we can check it over this parameters. So determination of biochemical variation in the drugs. So after completing that, so we should know what are all the biochemical characterization of the crude drugs having. So these things we should know. Okay, identification of deterioration due to treatment and storage to avoid the harmful effects getting after consumption of crude drugs. So before that, we should know about the entire biochemical and then importance. Is there any adulteration of the crude drugs? And finally, we should check the is there any harmful effects after consuming the drugs is present or not. So these are all the parameters we should know. So for that thing, we should know the importance. For that thing, we should know the evaluation of evaluation parameters. Okay. So after evaluation only we should know the importance. After if the evaluation part is complete, we can finalize. Okay, this drug is very much useful. So this drug is there is no side effects. This drug has this kind of biochemical characteristics, so we can consume it. So after completion of these evaluation parameters only, we should know the properties of the entire form of drug and benefits of the quality of evaluation. So what is the benefits of doing this evaluation means? Importance and accessibility of herbal product increased many folds. First one is importance. We should know the importance. Why we know the importance means, yeah, if you are doing the quality evaluation, then only we should know the chemical properties, physical properties of the drug. So we should know the importance. Okay, then for a strong linkage between fourth strong linkage between industries and national lab and quality assurance and batch to batch consistency, increased export potential, and desired effect. So, what in this slide conclude means if you are doing the quality evaluation of drugs, the first thing is we should know the biochemical characteristic of the exact drug, and then in company wise also they are using they are improving their sectors in quality assurance and batch to batch consistency and increased export potential and desired effect. These are all these many benefits of quality evaluations and the methods of drug evaluations. What are the methods which is includes in the drug evaluations? The drug evaluation is done by studying its various properties. So before evaluation, we should know the properties of the drug. So first one is organoleptic evaluation. The second one is microscopic evaluation. The third one is physical evaluation. Fourth one is chemical evaluation. The fifth one is analytical evaluation. And the final one is biological evaluation. The name itself it can explain what is organoleptic and microscopic, physical, chemical, analytical, biological. Okay. The first one is morphological evaluation. As we are morphology means the external features. Okay. Or organoleptic evaluation. The both are the same term. Okay. This refers to the drug evaluation, which means of our organs of sense and includes other sensory organs like odor, color, taste, and texture. What are all the these are the sensory organs. Okay, so it includes the study of morphology and other sensory characters. So in this evaluation, we are going to know the sensory characteristics of the specified drug. Okay. Study of gross morphology. It includes the visual examination of drug These drugs are classified into the following groups. What are all the visual examination? So we can see if you are taking on herbal plant, you can see which is the medicinal property. So, for example, if you take on a tulsi plant, the leaf has some aroma and the seeds has some aroma. So, this is the category of morphological and gross morphological evaluations. In this category, baths, underground structures, structures are nothing but our root, and next one is leaves and flowers, fruits, seeds, herbs. So these things we can see in our uh, so when coming to barks, it includes all the tissues in a woody stem outside the interfascicular cambium, which constitute to the drug. So can you see the yeah, we can see bark of any trees, but very few, not very few, some of the barks have medicinal properties. For example, in our home we can use what is that, cinnamon, that is a product of bark, and what we call it as and neem bark also we can use it as a medicinal properties, okay, medicine for some other diseases, okay. Bark collected from the trunk of branches or the trees and narrow strips, so narrow strips only we can collect. So during drying the drug, it undergoes unequal contractions and assumes different shapes. Example cinnamon, cinchona, ashoka, kurchi. These are all the some examples of bark. So here, this is slide shows the barks of different trees. First one is Arjuna, and the second one is cinnamon, the final one is cinchona. As I told you already, in our home we use a cinnamon. Okay, and underground structures. What are the underground structures? That is the plant which can store food via roots. So you can imagine uh, onion, garlic, ginger, and what is that? Turmeric. So these are the tuber plants. That means these can store food and using their roots. So rhizomes, roots, bulbs and tubers are the underground structure of plant. So they are swollen due to the storage of food material like carbohydrates and other chemicals. So why it look like a bulge a feature means they are due to the storage of food material, carbohydrate storage and other chemicals. So that purpose only, for that reason only, they are look like a storage, like uneven structures. Okay, examples ginger, turmeric, jatamansi. I don't know what is this, so you can refer and tell what is that. Okay, underground storage roots used as a drugs are ginger, turmeric. And next one is leaves. The shape, margin, base, apex and venation. Venation means this is okay, and branches like these are the venations, okay. That is different from each leaves, okay. See, here it is 2 3 4 5 6, okay. So that is a vary plant to plant, okay. The shape, margin, base, apex and venation of the leaves help in the identification of drugs. As I told you in the previous slide, identification is morphological and gross morphological. So this is the Morphological identification. This is the senna plant leaf. This is a eucalyptus leaf. This is a endaro leaf. And when coming to the flower, these are the reproductive organs of a plant. Yeah, we all know flower is the reproductive organ of the plant and possess different shapes. Okay, all flowers have different shapes, size, and even color also. Okay, fruits arise from the ovary and contain seeds. Fruits arise from the ovary of the plant, and also it contains seeds. They be globular, oblong, and ellipsoid in shape. It has different shapes. See the shapes: globular, ellipsoid. See, and this is the triple of fruit, which has n number of medicinal properties. Seeds. Seeds are developed from ovules in the carpels of the flowers. So seeds we all know which is derived from the carpels of the flowers. So they are characterized by the Hilum Nox vomica. This is the linseed and this is the Nox vomica seed. Study of sensory characters. So finally, this is the organoleptic study of either it is a crude drug, a plant, whatever it is. This is the organoleptic study. That is nothing but study of sensory characters, or in terms we can call it as an organoleptic study. Color, texture, odor, and taste are useful in the evaluation of drugs. First one is the color. Color. Some drugs are green in color when dried in shade, but they become pale and bleach when dried in sunlight. So you can see the difference. Some of the drugs are green color when dried in shade. So when you are going to dry in shade place, the color is different. But when we are going to shade in a sunlight, that could be a different color. Okay, due to the degradation process. Okay, this is the Terminalia cebula. This is a fresh seed and this is a dried one. So see the color changes. And odor. The odor of the drug may be either distinct or indistinct. The term used for the drugs are aromatic, balsamic, and spicy, etc. Mentha, clove are some of the examples for the drugs which have distinct odor. You can you smell these herbals? Mint, mint, clove. Yeah, everyone can smell. So these aromas of this drug have distinct odor. Okay, and when coming to the taste, the drugs may be evaluated by drugs taste also. The taste may be saline, sour, salty, sweet, bitter, alkaline, etc. So the substance without taste are regarded as tasteless. So if the drug they don't have any taste, which is called as tasteless. Example, ginger, capsicum. So all of you can taste these two. Texture. Sometimes drugs can be examined by their consistency, texture, and nature of the fracture. Example, colocynth can be compressed easily since its parenchyma is loose. So, what is the texture means? Whether it is tightly packed or loosely packed. So, accordingly, can be considered. So first one is a morphological characteristics. The second one is microscopic and anatomical evaluation. So this is we can see in next class.